Good morning, everyone. I'm joining you again today from Park City, Utah, where I'm here for a conference for coaching. And I wanted to talk to you a little bit about signal transduction today. I also wanted to show you the view from my room. And this morning I went out to a brand new coffee shop and I was thinking a lot about signal transduction because I ordered a latte. I was like, oh my gosh, thinking about the change in my body from signaling the hungry signal to signaling fed. And that got me thinking a lot about signal transduction. Of course, the caffeine in the latte also got me thinking about how does caffeine affect signal transduction and perpetuation of signals as they are amplified. So that's where we're going to turn our attention today, is to signal transduction and looking at how not only integral membrane proteins can play a role in transport, as we've been seeing, but now also looking at how they can play a role in signal transduction. So we'll come back inside and sit down and let's do a little bit of writing here. To kick off our coverage of signal transduction, I've begun by writing the general mechanism by which many signaling molecules get their message from one cell to another. So let's just jot down that this is a generalized signal transduction pathway, and we'll look at how signals are amplified as they reach a cell. Now, the nature of these signals can be very diverse, and of course, signaling occurs across all domains of life. Those of you who have studied microbiology recall that many bacterial cells get signals from other molecules that lead them in many cases to move to a particular site. So that is to say, a signal that a cell might receive might in fact be a chemotactic signal saying to another cell, hey, get over here, right? Leading to the movement of that cell. So the molecule that enacts that we always call a ligand. A ligand is going to be the first messenger in signaling, and that ligand may be bringing a variety of messages. We often actually call this ligand the voice of the cell. So a first messenger is carrying a vocal message, if you will, from one cell to another, telling it something. Now, this messenger might be a hormone, and that was the type of messenger that I was thinking about this morning as I went from signaling a hungry state to signaling a fed state. And so, for example, the hormone signal might be the hormone called glucagon, and we know glucagon signals hungry. So when I woke up this morning, my hungry signal was going like crazy. We may also see signals or first messengers that are neurotransmitters that may signal some sort of neural message at a synapse. We may see those first messengers that are growth factors. The growth factors may be signaling need for cell proliferation or differentiation and proliferation. So there may be a variety of messages that may be heard from between one cell to another cell. And let's look at now what happens when a ligand reaches its target cell and it binds to the receptor. Notice that the receptor is a transmembrane protein that receives that signal. This forms a receptor-ligand complex, and that will then affect a change within the cell. You can think about the sort of molecular changes that would be going on here. When a ligand binds to a receptor, that receptor site will very specifically accommodate that ligand, and it will kick off changes in the amino acids that line that particular binding site. That will eventually lead to a change, and within the cell it will activate the next molecule, and in this kind of signal transducing pathway, this is actually the sort of notorious molecule in the pathway, because this is the transducer. So the transducer becomes activated, and this transducer now carries the signal to the next molecule, also a transmembrane protein, an enzyme called an effector. This effector catalyzes a reaction in which a second messenger is created. So here we've gone from an extracellular first messenger that was triggering a change from the outside to an intracellular second messenger. And the second messenger may vary in nature. It may be something such as cyclic GMP or cyclic AMP. Cyclic AMP is one of the most common, and I've drawn it over here in the corner. Cyclic AMP is similar. You're probably noticing that that looks pretty similar to ATP in structure, except for that notice instead of having 3 phosphate groups on it, it actually only has a single phosphate. So adenosine monophosphate, only 1 phosphoryl group on that molecule. But notice further that it's cyclized. That is, that the oxygen bound to the carbon number 5 in the ribose sugar is also then bound to the phosphorus group, or the phosphorus atom within the phosphoryl group, which is also then bound to the oxygen on the number 2 carbon in the sugar ring. So we're recognizing a cyclization there to form cyclic AMP, a very, very common second messenger. These second messengers may also be things like nitrous oxide. They may be things like PIP2. We'll see that being an important second messenger in the rest and digest signal. So there are a variety of different second messengers, but in all cases, they lead to some sort of cellular response. Now, if the signal being sent is a signal that is signaling hungry, then, and perhaps this particular ligand is binding to your liver, perhaps the cellular response within the liver cell in the hepatocyte is to say, hey, break down some glycogen and ship out some glucose to those tissues that are crying out for a little bit of that fuel molecule. So that could be the cellular response that happens to be enacted due to that second messenger. One feature that we see being conserved in signal transduction is the principle of amplification. That is to say that when we get an initial signal, that initial signal is amplified. So to be more specific about what I mean there, a particular receptor ligand complex might activate more than one transducer. That transducer, once activated, might activate more than one effector enzyme, and the second messenger might enact changes on more than one target molecule at that point. So we get amplification, right? One signal goes to being 2, and we get this, what we might think of as exponential growth of the signal or amplification of the signal due to, for example, one receptor ligand complex activating more than one transducer, and so on forth. So we might jot down amplification is an important feature of signal transduction. A single ligand receptor complex may interact with multiple transducers, which can activate several molecules of effector enzyme. Again, this is termed a signaling cascade. And I tell you what, there's not much that can get more complex than signaling cascades. Man, it can really, really get deep really fast. But I want to also note that although this is a very common means by which a ligand enacts its intracellular eventual intracellular change, this is not the only type of signaling that we can see from even hormones. For example, those of us who receive frequent and sometimes unwanted signaling from estrogen, estrogen actually binds to genes. And if I can feel it, but I can't spell it, right? So estrogen commonly binds upstream of genes, which means that it actually itself is capable of weaseling across the lipid bilayer and into the nuclear, through the nuclear envelope and onto genes, where it sits on top of gene response elements called estrogen response elements, or EREs. So this is another way in which hormonal signals can be heard and felt by a cell that is receiving that signal. I hope that this gives you the broad overview of signal transduction so that we can look more at the details of one particular signaling pathway.